We're going inside.
Exactly. We're embarking on a true, detailed tour of the fundamental unit of life. We're really diving deep into this, well, massive stack of material we have. It covers everything from the foundational theories, the basics, right up to the cutting-edge microscopy that lets us actually see these things happening.
It's easy to think of a cell as just this tiny, simple blob, isn't it?
Hmm.
But the truth is, every single cell is like a highly integrated, incredibly specialized factory.
A city unto itself, almost.
That's a good way to put it. Complete with command centers, complex communication networks, super efficient transport systems. Our mission today really is to pull out the most important nuggets from all this information.
Yeah.
To help us understand the deep architecture of these tiny machines. And crucially, why all that complexity is just absolutely essential for life as we know it.
Absolutely. And to really get that complexity, I think we have to start right at the beginning with the rules, the foundational rules that define life at this scale.
Right. The bedrock is the cell theory.
First put together by Schleiden and Schwann, and then Virchow added that key piece later in the mid-19th century.
And that theory gives us three, well, non-negotiable pillars. First, all living things, bacteria, whales, you, me, everything is made of cells.
Okay, pillar one.
Pillar two. The cell is the most basic, the functional unit of life. It's the smallest thing you can point to and say, that's alive.
Fundamental unit.
And third, Virchow's crucial addition. All cells come from pre-existing cells. Omnicellula, ecellular.
So no spontaneous generation.
Exactly. Life doesn't just pop into existence. It's continuous. It's inherited, passed down.
So we've got this perfect, self-contained, efficient little machine. Okay, let's unpack this a bit. If cells are so great, so effective, and usually bigger means more complex, maybe more efficient, why aren't large organisms just one mingle enormous cell?
Huh. Yeah, why do we bother with trillions of them?
Right. Why the logistical nightmare of being multicellular?
It's a fantastic question, and the answer is pure physics. It comes down to geometry. It's called the surface area to volume ratio constraint, or SAV.
SAV.
This physical principle puts a hard upper limit on how big any single cell can get and still, work.
I get the concept, sort of, but maybe walk us through the physical implications. The surface area is like the cell's connection to the outside world.
Exactly that. The cell membrane, the surface, is the gateway. It's the only place nutrients, oxygen, signals can come in. And it's the only place waste can get out.
The loading dock.
The loading dock, yeah. The volume, though, that's the inside. That's where all the work happens, the metabolism, energy use, production. And here's the catch. As a cell gets bigger, its volume increases much, much faster than its surface area does.
Ah, okay, so the internal factory floor gets huge, but the loading dock doesn't grow nearly as fast.
You've got it. The internal demand, the metabolic need, quickly outstrips the capacity of the membrane to supply it or get rid of the garbage.
So it basically suffocates or poisons itself.
Pretty much. We've got a great numerical example here. Imagine a tiny cell, a cube, just 1 micrometer on each side. Tiny.
Okay.
It has a fantastic SAV ratio, 6. Every bit of its inside is really close to the surface. Super efficient transport.
Makes sense.
Now, just scale it up 10 times, make it a 10 centimeter cube. Still microscopic, right?
Yeah, still tiny to us.
But its SAV ratio plummets. It drops all the way down to 0.6.
Wow, that's a huge drop.
It is. The volume has ballooned, but the surface area just hasn't kept pace. It can't get nutrients in fast enough to feed that big interior, and it can't get waste out efficiently. It starves.
That's actually really elegant. It explains so much about biological design. Not just why we're multicellular, but why some larger cells have such weird shapes.
That's a really key point. Biology is clever. It finds ways around the constraint without actually breaking the physical law. Think about the cells lining your gut or your lungs.
Epithelial cells.
Right. They're often really flat or long and thin. That maximizes their surface area compared to their volume.
And what about things like microvilli, those little finger-like things you see on gut cells? They seem like nature cheating the system.
Microvilli are the perfect cheat. They're these tiny projections that massively increase the cell's effective surface area without really changing the overall volume much at all.
So more loading dock space.
Tons more. Absolutely essential in places like the small intestine, where you need every square nanometer you can get for absorbing nutrients. So it's a physical limit, but life adapts, it folds, it flattens, it projects. And for us, well, it means we have to be this community of trillions of small optimized units, all playing by these geometric rules. Okay, so we understand the size issue. Now we need to look at this really deep evolutionary split in how cells are built. Life is divided into three main domains: bacteria, archaea, and eukarya.
And the first two, bacteria and archaea, the prokaryotes, the simpler design, right? The ones that ruled the early Earth.
That's right. They're generally the small guys, maybe 1 to 10 micrometers across, and their defining feature is, well, what they lack.
Right, here's where it gets really interesting. Their DNA isn't neatly wrapped up in a nucleus. It's usually a single circular chromosome, just kind of clustered in a region they call the nucleoid.
Yeah, no membrane around it. And often they have extra little circles of DNA too, called plasmids. These can carry useful genes like antibiotic resistance.
Uh-huh.
But the big difference, the defining one, is no internal membrane-bound compartments. No nucleus, no mitochondria, no ER, none of that internal subdivision.
Simple, fast, efficient for their size.
Exactly. But even within prokaryotes, there are important distinctions.
Okay, give us the specifics. Bacteria versus archaea.
Okay, bacteria. Their hallmark is a cell wall made mostly of peptidoglycan. It's this unique polymer, a mesh of sugars and amino acids. Gives them shape and protection.
Peptidoglycan.
Got it. Archaea, on the other hand, they're often the extremophiles, living in boiling springs or super salty lakes. They are fundamentally different. Their cell membranes use unique types of lipids that help them survive those harsh conditions.
And their walls.
No peptidoglycan. Never. They might use other things like proteins or something called pseudomurane, but not the same stuff as bacteria.
Okay, distinct chemistries. And what about their internal machinery? Like the protein factories? Ribosome.
Ah, yes, the ribosomes, another key difference. Prokaryotic ribosomes are a bit smaller. They're called 70S ribosomes, made up of a 30S and a 50S subunit. Remember those numbers, they become important later.
70S, 30S, 50S.
And prokaryotes often have external bits too. Flagella for swimming around, pili, which are shorter, maybe for sticking to surfaces or for exchanging those plasmids, that's conjugation, and sometimes a sticky outer layer, a capsule for extra protection.
Okay, so that's the prokaryotic world. Now let's cross the dividing line to the eukaryotes. That's us, animals, plants, fungi, protists.
Right, and the difference is huge, both in size and organization. Eukaryotes are typically much larger, maybe 10 to 100 micrometers.
10 to 100 times bigger.
Easily. And the defining architectural leap is compartmentalization. They have a true nucleus wrapped in a membrane holding multiple linear chromosomes. And they have a whole suite of other membrane-bound organelles, tiny organs, really each doing a specific job.
The internal departments we talked about.
Yeah.
And their ribosomes are different too.
Yep. Slightly larger, slightly different shape. Eukaryotic ribosomes are 80S, made of a 40S and a 60S subunit.
80S, 40S, 60S.
Okay. And this compartmentalization, this internal division of labor, is the secret to their success. By separating different chemical reactions into different rooms, the cell can do things that would be impossible otherwise.
Like keeping toxic processes contained.
Exactly. Or maintaining different environments, like the highly acidic interior of a lysosome. It allows for massive increases in biochemical efficiency compared to the sort of one-room studio apartment of a prokaryote.
Now, the really crucial takeaway here, the so what for us, is that these structural differences, peptidoglycan walls, 70S versus 80S ribosomes, they aren't just interesting biological facts. They are the absolute foundation of modern medicine, specifically antibiotics.
This is it. This is why antibiotics work. They exploit these differences. Take penicillin, the classic example.
Right.
It works because it specifically attacks the enzyme that builds that peptidoglycan cell wall.
Which our cells don't have.
Exactly. Our eukaryotic cells lack peptidoglycan entirely. So penicillin messes up the bacterial wall synthesis. The bacterium can't handle its internal pressure, and poof, it bursts. Our cells are totally unharmed. It's a brilliantly selective weapon.
And the same idea applies to targeting their ribosomes.
Absolutely. Drugs like tetracyclines or aminoglycosides, they are designed to specifically bind to the smaller 30S subunit of the prokaryotic 70S ribosome,
Gumming up the works.
totally shutting down their protein production. Our 80S ribosomes, with their different 40S small subunit, aren't affected nearly as much. The drug basically ignores our protein factories while crippling the bacterias. Understanding this molecular architecture isn't just academic, it's literally life and death when you're fighting an infection.
Okay, so eukaryotic success comes from compartmentalization. We think of it like a highly organized factory. Let's start our tour right at the top, the control room, the nucleus.
The nucleus.
Oh.
It's the vault holding the genetic blueprint, the DNA. It's protected by a double membrane, the nuclear envelope.
Cell membrane, okay.
And that envelope isn't sealed tight. It's dotted with nuclear pores. These aren't just simple holes. They're incredibly complex protein structures that act like very strict security checkpoints. They control everything that goes in or out.
Letting RNA out, letting proteins needed for DNA replication in.
Precisely. Inside, the DNA isn't just floating around, it's tightly packaged with proteins. This DNA protein complex is called chromatin.
And this command center has to copy that blueprint with unbelievable accuracy during cell division. The details on the machinery for DNA replication are pretty amazing. It's not just unzipping and copying, is it?
Oh, not at all. It's a molecular ballet, a carefully orchestrated process involving a whole team of specialized enzymes.
Let's name some names.
Okay, first you need helicase. That's the enzyme that physically unwinds and separates the two strands of the DNA double helix, like a zipper pull.
Okay, strands separated.
Then you have primose. It lays down a short little piece of RNA called a primer. Why? Because the main building enzyme, DNA polymerase, can't start from scratch. It needs something to add on to.
It needs a starting block.
Exactly. Once the primer is there, DNA polymerase takes over. It zooms along the template strand, adding the correct DNA nucleotides one by one, building the new complementary strand.
Now, that unwinding process sounds like it would cause problems. If you untwist one part of a rope, the rest gets tighter, right?
You're absolutely right. Imagine untwisting a tightly wound rubber band. The tension downstream builds up incredibly quickly. The DNA would get supercoiled and eventually just snap.
So how does the cell deal with that?
With another amazing enzyme called topoisomerase, it works ahead of the replication fork. It nicks one or both DNA strands, lets the helix swivel around to release that built-up tension, and then instantly seals the break.
Wow, like a molecular pressure relief valve?
Perfectly put. Without topoisomerase, replication would grind to a halt almost immediately.
Okay, so polymerase builds the new strands, but I remember something about one strand being made in pieces.
Yes, the lagging strand.
Mm.
Because DNA polymerase can only build in one direction, one of the new strands is synthesized continuously, but the other has to be made in short fragments called Okazaki fragments.
Okay.
And that's where our final key player comes in, ligase. DNA ligase acts like molecular glue. It goes along and joins those Okazaki fragments together, sealing the gaps in the sugar phosphate backbone to create a single continuous DNA strand.
Helicase, primase, polymerase, topoisomerase, ligase. Quite the team.
An essential team for life.
But the nucleus isn't just managing the DNA blueprint. It's also making the machinery for the protein assembly line itself, right? In that dense spot called the nucleolus.
Exactly. The nucleolus is like a factory within the factory. Its main job is churning out ribosomal RNA, rRNA, and assembling the ribosomal subunits, those 40S and 60S pieces we mentioned.
So it builds the parts for the protein factories.
It does. Those subunits are then shipped out through the nuclear pores into the cytoplasm, where they come together to form functional ribosomes ready for action.
Okay, so the ribosome subunits are ready. Let's move out to the cell's main assembly lines. The ribosomes themselves and the endoplasmic reticulum, the ER.
Right. Ribosomes are the actual protein builders. They read the messenger RNA instructions and link amino acids together. And they're unique. They don't have a membrane around them.
And their location matters, doesn't it? Free versus bound.
Absolutely critical distinction. You have free ribosomes just floating around in the cytosol, the main cell fluid. They make proteins that are destined to function right there in the cytosol.
Like enzymes for metabolism or structural protein.
Exactly. Then you have bound ribosomes. These guys are physically stuck to the surface of the endoplasmic reticulum.
Giving that rough appearance.
Precisely, hence the rough ER, or RER. These ribosomes are making proteins that have different destinations. They might be secreted out of the cell entirely, or embedded in one of the cell's membranes, or sent to another organelle like the Golgi or lysosomes.
So the RER is the first stop for these non-cytosolic proteins. What happens there?
The RER is all about processing these newly made proteins. As the protein chain is synthesized, it's threaded into the RER lumen, the space inside. And there, several key things happen. First, protein folding.
Getting that complex 3D shape right.
Crucial. And it's often helped by other proteins called chaperones. They guide the folding process, prevent mistakes. The RER also has quality control. If a protein doesn't fold correctly, it's usually tagged and sent off for destruction.
And modifications happen too, like adding sugars.
Yes, that's initial glycosylation. Sugar chains are often added to the protein right as it's being made and entering the RER. These sugar tags can be important for stability or for signaling where the protein needs to go later.
Okay, that's the rough ER, studied with ribosomes, focused on proteins. What about the smooth ER, the SER? No ribosomes there.
Right, it looks smooth. And its functions are quite different, mostly related to lipids and detoxification. First, it's the main site for making lipids, phospholipids for membranes, steroid hormones, things like that.
Okay, lipid synthesis.
Second, it's a major detoxification center, especially in liver cells. It's packed with enzymes, particularly the famous cytochrome P450 family.
Ah, yes, those are involved in breaking down drugs and toxins.
They are. They modify these substances, often making them more water soluble so the body can get rid of them more easily.
And the third big job for the SCR. Tell me about calcium.
Yes. This is incredibly important and sometimes overlooked. The SCR acts as the cell's main storage tank for calcium ions, call.
Why is storing calcium so important?
Because calcium ions are critical signaling molecules inside the cell. A rapid change in calcium concentration triggers all sorts of events: muscle contraction, neurotransmitter release, hormone secretion. The SCR holds on to this calcium until it gets the signal to release it.
Like in muscle cells.
Exactly. In muscle cells, the SCR is so specialized for calcium storage and release, it gets its own name, the sarcoplasmic reticulum. A nerve signal triggers calcium release from the SR, and that calcium flood causes the muscle fiber to contract. It's a beautiful example of how compartmentalizing something, in this case, calcium enables a rapid, specific response.
Okay, so proteins and lipids are made and get some initial processing in the ER. But they're not ready for their final jobs yet, are they? They need more sorting, modifying, packaging.
That's right. They're like raw materials or partially finished goods. They need to go to the finishing and distribution center. And that's the Golgi apparatus.
With the cell's post office.
An excellent analogy. The Golgi is usually seen as a stack of flattened, membrane-bound sacs called cisternae.
And it has directionality, right? A receiving side and a shipping side.
It does. Vesicles bud off from the ER carrying proteins and lipids, and they fuse with the cis face of the receiving dock.
Okay.
The cargo then moves progressively through the stack from cisterna to cisterna towards the transface, the shipping department, and as it moves, it gets modified.
What kind of modifications happen in the Golgi?
Further refinement of those sugar chains added in the ER is a big one. Glycoproteins get processed, phosphate groups might be added, sulfate groups. These modifications act like molecular shipping labels or ZIP codes.
Determining the final destination.
Precisely. One specific tag might signal that this protein is destined for a lysosome. Another might signal it should be packaged into a vesicle that will fuse with the outer cell membrane for secretion. The Golgi sorts it all out and then packages the finished products into new vesicles that bud off from the transface. It even makes the lysosomes themselves.
Okay, speaking of lysosomes, let's move to the cell's waste management and recycling systems. Lysosomes and peroxisomes. Lysosomes, first you call them the digestive system, like a controlled acid bomb.
That's pretty accurate, actually. Lysosomes are small vesicles filled with powerful hydrolytic enzymes that break down molecules using water. And critically, they maintain a very acidic internal environment around pH 5.
Why the low pH?
Because those hydrolytic enzymes work best in acidic conditions. The rest of the cell, the cytosol, is neutral, around pH 7.2. So if a lysosome accidentally leaks, its enzymes won't do much damage outside because they're not in their optimal environment. It's a safety feature.
Clever. And what kind of destructive enzymes are in this cocktail?
A whole arsenal. You've got proteases to chop up proteins, lipases for lipids, nucleases for DNA and RNA, glycosidases for complex carbohydrates. Basically, they can digest almost any biological macromolecule.
And what do they digest? Stuff coming in from outside?
Yes. They fuse with vesicles formed during phagocytosis cell eating to digest captured bacteria or food particles. But just as importantly, they handle internal cleanup through a process called autophagy.
Autophagy. Self-eating.
Literally. The cell can engulf old, worn-out organelles like a damaged mitochondrion or clumps of misfolded proteins within a membrane and then fuse that package with a lysosome. The lysosome digests the contents, recycling the valuable molecular building blocks back into the cytosol for reuse. It's essential for cellular health and maintenance.
And when this recycling system breaks down, the consequences are severe, right? Leading to specific diseases.
Absolutely. If just one of those crucial lysosomal enzymes is missing or defective due to a genetic mutation, the substance it's supposed to break down starts to accumulate inside the lysosomes. This leads to a group of conditions called lysosomal storage diseases.
Like Tay-Sachs.
Tay-Sachs disease is a tragic, classic example. A deficiency in an enzyme called hexosaminidase means a specific lipid GM2 ganglioside can't be broken down. It builds up, particularly in nerve cells, causing massive swelling and progressive neurological damage, ultimately leading to death, usually in early childhood. It really highlights how vital every single component of the system is.
Okay, so lysosomes are the main breakdown and recycling center. What about peroxisomes? They sound different, maybe like the hazardous waste disposal unit.
That's a really good way to think about them. Peroxisomes handle specific types of metabolic reactions, many of which involve oxidation and critically produce or consume hydrogen peroxide.
Hydrogen peroxide, HERO. That's
Yeah.
pretty reactive stuff.
Very reactive, potentially damaging. Peroxisomes contain enzymes called oxidases that use oxygen to break down molecules like fatty acids and amino acids. This process generates hydrogen peroxide as a byproduct.
Okay.
But peroxisomes are also packed with enormous amounts of another enzyme called catalase. Catalase takes that dangerous hydrogen peroxide and immediately breaks it down into harmless water and oxygen.
So they generate a hazard but also contain the cleanup crew.
Exactly. It's self-contained hazardous waste processing. Besides breaking down fatty acids, a process called beta oxidation, which actually generates some energy intermediates. Peroxisomes are also crucial for detoxifying certain substances.
Like alcohol.
Yes, in liver cells, peroxisomes help break down ethanol into acetaldehyde. So their jobs include specific metabolic pathways, detoxification, and importantly, neutralizing that reactive hydrogen peroxide, essential damage control.
All right. Finally, in our organelle tour, let's get to the power plants and, well, bit of deep history. Mitochondria and chloroplasts. Their very existence points to one of the coolest ideas in biology, endosymbiotic theory.
It really does. The theory proposes that both mitochondria and chloroplasts were once free-living bacteria that got swallowed up by a larger ancient host cell billions of years ago.
Swallowed but not digested.
Exactly. Instead of being eaten, they formed a symbiotic relationship. They lived together, benefiting each other. The engulfed bacterium provided some advantage, maybe efficient energy production or photosynthesis, and the host cell provided protection and resources. Over time, they became completely dependent on each other. Mitochondria are thought to have descended from an ancient type of bacteria called an alpha proteobacterium.
And the evidence for this ancient merger is still visible inside them today.
Oh, absolutely. The evidence is compelling. First, both have a double membrane. The inner membrane resembles the original bacterial membrane, and the outer one likely came from the host cell's membrane when it engulfed the bacterium.
Okay, double membrane. What else?
Crucially, they have their own DNA. It's separate from the main DNA in the nucleus, and it's typically a small circular chromosome, just like in bacteria. And they have their own ribosomes to make some of their own proteins. And guess what kind of ribosome?
Let me guess. 70S.
Yeah,
Like bacteria.
bingo. They have smaller 70S ribosomes just like their prokaryotic ancestors, not the 80S ribosomes found in the host cell's cytoplasm. It's like finding ancient bacterial machinery still running inside our own cells.
We usually think of mitochondria just as the powerhouses, making ATP through cellular respiration. But they have another critical role, don't they? In cell life and death.
Yes, it's a vital dual role. They are the primary sites of energy conversion, absolutely, but they are also key regulators of programmed cell death, or apoptosis.
How do they do that?
If a cell receives signals indicating severe damage or that it's no longer needed, the mitochondria can initiate the self-destruct sequence. They release a protein called cytochrome C from the space between their membranes into the cytosol.
And cytochrome C is the trigger.
It's a key trigger. Once in the cytosol, it activates a cascade of destructive enzymes called caspases, the cell's executioners. This ensures the cell dies cleanly and efficiently without spilling its contents and causing inflammation in the surrounding tissue. So mitochondria are central to both keeping the cell alive with energy and ensuring it dies properly when necessary.
And in plants and algae, we have the other endosymbiont, chloroplasts.
Right, the sites of photosynthesis. They share the same endosymbiotic hallmarks. Double membrane, their own circular DNA, their own 70S ribosomes.
And structurally, they have those internal stacks.
Yes, their internal structure is dominated by intricate membrane systems. You have flattened sacs called thylakoids. These contain chlorophyll and the machinery for the light-dependent reactions capturing light energy. Thylakoids are often stacked up into structures called grana.
And the fluid around them.
That's the stroma. The stroma is the fluid-filled space inside the inner membrane but outside the thylakoids. It contains the enzymes for the Calvin cycle, the reactions that take sero from the air and use the energy captured by the thylakoids to build sugars. Again, perfect compartmentalization. Light reactions in the thylakoids, sugar building in the stromal.
Okay, so we've toured the organelles, the specialized department. But a cell isn't just a bag of organelles. It needs structure. It needs to move things around. It needs to interact. Let's talk about the cell's internal scaffolding and highway system, the cytoskeleton.
The cytoskeleton is so much more than just passive scaffolding. It's incredibly dynamic. It provides shape, yes, but it's also involved in movement, both the cell moving itself and moving things within the cell. It's made up of three main types of protein filaments.
Okay, let's start with the smallest ones, microfilaments.
These are thin, solid rods made primarily of the protein actin. Actin filaments are really dynamic. They assemble and disassemble rapidly. They're crucial for cell shape changes, muscle contraction along with myosin, cell crawling, forming those microvilli we talked about, and pinching the cell in two during cell division, a process called cytokinesis.
So involved in movement and shape changes. Then we have the intermediate filaments.
Right. These are more like ropes than rods, providing tensile strength resistance to stretching and mechanical stress. They're much more stable than actin filaments. Think of keratin in your skin cells, which makes skin tough, or the nuclear lamins that form a supportive mesh just inside the nuclear envelope. They're the structural anchors.
Holding things together under stress. And the third type, the largest ones, microtubules.
Microtubules are hollow tubes made of the protein tubulin. These are like the cell's major highways or girders. They're much more rigid than actin filaments. They radiate out from a central organizing center.
And they have several big jobs.
Absolutely. They form the mitotic spindle, the crucial structure that segregates chromosomes accurately during cell division. They're the core structural component of cilia and flagella, the whip-like appendages used for movement. And, very importantly, they act as tracks for intracellular transport.
So organelles and vesicles move along these microtubule tracks.
Exactly. But the tracks themselves don't provide the movement. You need engines. And that's where the motor proteins come in.
The molecular motors. This is amazing stuff.
It really is. There are two main families that walk along microtubules, kinesin and dynein. They use energy from ATP hydrolysis to physically step along the microtubule, carrying cargo attached to them, vesicles, organelles, even large protein complexes.
And they go in different directions.
Generally, yes. Kinesin motors typically move cargo away from the cell center out towards the periphery. This is called anterograde transport. Dynein motors usually move cargo towards the cell center or retrograde transport.
This must be incredibly important in nerve cells with those long axons.
Absolutely critical. Imagine needing to transport neurotransmitter-filled vesicles from the cell body all the way down an axon that could be a meter long. It all depends on kinesin motors chugging along microtubule tracks. And bringing back old components relies on dynein. It's a constant, highly regulated traffic system.
And like the differences between prokaryotes and eukaryotes, this cytoskeleton machinery can also be a target for medicine, especially cancer treatment.
Yes, because rapidly dividing cells, like cancer cells, rely heavily on the microtubule dynamics of the mitotic spindle to divide. So drugs that interfere with microtubules are powerful anti-cancer agents.
Like Taxol.
Exactly. Paclitaxel, or Taxol, works by stabilizing microtubules. It prevents them from breaking down, which sounds good, but the spindle needs to be dynamic. It needs to assemble and disassemble. By freezing it, paclitaxel halts cell division, preferentially killing rapidly dividing cancer cells. Other drugs, like vincristine, prevent microtubule assembly.
And problems with the cytoskeleton are linked to other diseases too, like Alzheimer's.
Yes, unfortunately. In Alzheimer's disease, a protein called tau, which normally helps stabilize microtubules in neurons, becomes abnormally modified and forms tangles inside the cells. This disrupts the microtubule tracks, impairing transport within the neuron, which contributes significantly to neuronal dysfunction and death.
Okay, let's step outside the cell now. Cells don't exist in isolation. They have external structures and they need to connect with their neighbors. Let's talk extracellular components and cell junctions.
Right. The outside environment is crucial. We already mentioned some external structures. Plant cells have a rigid cell wall made mainly of cellulose. Fungi use chitin. Bacteria use peptidoglycan.
But animal cells don't have a rigid wall like that. What do they have?
Animal cells secrete and surround themselves with an intricate network called the extracellular matrix, or ECM. It's not just passive packing material, it's dynamic and involved in structure, adhesion, and signaling.
What's it made of?
It's a complex mix, but key components include strong, fibrous proteins like collagen, which provides tensile strength, and elastin, which provides elasticity or recoil, like in your skin or blood vessels. There are also adhesive glycoproteins like fibronectin, and importantly, cells connect to this matrix via transmembrane proteins called integrins. Integrins physically link the internal cytoskeleton to the external ECM, bridging the inside and outside worlds.
So cells are anchored to this matrix. But how do they connect to each other to form tissues? That requires specialized cell junctions.
Exactly. Tissues wouldn't hold together without them. We can group junctions based on their main function. First, you need sealing junctions.
Like making a sheet of cells waterproof.
Precisely. Tight junctions do this. They form a belt-like seal completely around each cell in an epithelial sheet, like the lining of your gut or bladder. They prevent molecules and bacteria from leaking through the gaps between the cells. They force substances to go through the cells, allowing for regulated transport.
Okay, sealed up. What about holding cells together strongly? Anchoring junctions.
Yes, several types provide mechanical strength. Adherens junctions also form a belt around cells, usually just below the tight junctions. They use proteins called cadherins that link to the actin microfilaments inside adjacent cells. They connect the actin cytoskeletons of neighboring cells.
Cadherins linking actin. Got it.
Then you have desmosomes. These are like spot welds providing very strong adhesion. They also use cadherins, but these cadherins link to the much stronger intermediate filaments, like keratin, inside the cells. This creates a tissue-wide network of intermediate filaments, giving the tissue great resistance to mechanical stress, like in your skin or heart muscle.
Cadherins linking intermediate filaments. Stronger connection.
And one more anchor type. Hemidesmosomes. Hemi means half. These look like half a desmosome, and they anchor the intermediate filaments of a cell not to another cell, but to the underlying extracellular matrix, the basement membrane, using integrins instead of cadherins. They staple the cell layer down.
And the failure of these anchoring systems, particularly involving cadherins, is implicated in cancer spreading.
Yes, this is hugely significant. Many epithelial cancers undergo a process called the epithelial mesenchymal transition, or EMT, which allows them to become invasive and metastasize. A key step in EMT is often the loss or inactivation of E-cadherin, the main cadherin in adherens junctions.
So losing the glue lets the cells break free.
Essentially, yes. When E-cadherin function is lost, the cells become less tightly adhered to each other, more mobile, and can detach from the primary tumor, invade surrounding tissues, and potentially travel to distant sites to form metastases. It's a critical molecular switch in cancer progression.
Okay, sealing and anchoring. What about direct communication between cells?
For direct cytoplasmic sharing, we have gap junctions in animal cells. These are channels formed by proteins called connexons that span the membranes of two adjacent cells.
Creating a little tunnel between them.
Exactly. Small molecules and ions, like calcium or cyclic AMP, can pass directly from the cytoplasm of one cell to the next through these channels. This allows cells in a tissue to coordinate their activity rapidly, like the synchronized contractions of heart muscle cells or coordinated responses in developing tissues.
And plants have something similar, even with their thick walls.
They do. Plants have plasmodesmata. These are membrane-lined channels that pass directly through the cell walls, connecting the cytoplasm of adjacent plant cells. They serve a similar function to gap junctions, allowing communication and transport between cells.
And of course, nerve cells have their own specialized communication junction.
The synapse. This is highly specialized for rapid directional signaling. It involves the release of chemical signals neurotransmitters from one neuron across a tiny gap, the synaptic cleft, to receptors on the next neuron. It's the basis of all nervous system function.
We've journeyed through structures, ribosomes, motor proteins, nuclear pores, that are incredibly tiny, often far smaller than the wavelength of visible light. So the big question is, how on earth do we know all this? How do scientists actually see things that are nanometers across?
That's the crucial point, isn't it? This entire tour is only possible because of incredible advances in microscopy. Our starting point, basic light microscopy, is great, but it hits a fundamental limit. Because of the wavelength of light, you just can't resolve details much smaller than about 200 nanometers.
So you can see cells, maybe the nucleus, but not the really fine details inside.
Right. To see organelles and smaller structures clearly, we needed ways to make specific parts stand out.
That led to fluorescence microscopy.
Using glowing tags. Exactly. You can attach fluorescent molecules either using antibodies that bind to specific proteins or by genetically engineering cells to produce proteins fused to something like green fluorescent protein, GFP, to label the structure you're interested in, say mitochondria or the cytoskeleton. Then you shine specific wavelengths of light on it, and only your tagged structure glows brightly against a dark background.
And confocal microscopy takes that a step further.
It does. Confocal uses lasers to illuminate just a tiny spot in the sample at a time and a pinhole aperture to block out any out-of-focus fluorescent light coming from above or below that spot. By scanning the laser across the sample, you can build up a sharp optical slice.
And stack those slices together?
Yep, stack them up digitally to create incredibly detailed 3D reconstructions of cells and tissues. It was a huge leap forward.
But even with fluorescence, that 200 nanometer diffraction limit was still a barrier for a long time, wasn't it? Until the development of super-resolution microscopy.
That's right. Techniques like STED, PALM, and STORM came along relatively recently and managed to smash through that theoretical limit. They use various clever tricks like switching fluorescent molecules on and off selectively, or precisely locating single molecules to achieve resolutions down to tens of nanometers, sometimes even better.
So now we can actually see things like individual protein movements within a membrane.
We can. It's opened up a whole new level of understanding of molecular dynamics within living cells.
But to get the absolute highest resolution, the finest details of ultrastructure, we need to dish light altogether and use electrons, right? Electron microscopy.
Exactly. Electrons have much shorter wavelengths than visible light, allowing for vastly higher resolution, down to the sub-nanometer scale in some cases. There are two main types, TEM, or transmission electron microscopy.
That's looking through the sample.
Right. You have to prepare an extremely thin slice of your sample, often stained with heavy metals to increase contrast. Then you shoot a beam of electrons through it. Where the electrons pass through easily, it looks bright. Where they're scattered by the heavy metal skeins, it looks dark. This gives you incredibly detailed cross-sectional views of the internal machinery. You can see the membranes of the ER, the folds, cristae inside mitochondria, individual ribosomes.
And the other type? SEM.
SC, or scanning electron microscopy. This gives you amazing surface views. You coat the sample surface with a thin layer of metal, then scan an electron beam across it. The electrons bouncing off the surface are detected, generating a 3D-like image of the sample's topography. Great for looking at the outside of cells or things like pollen grains or insects.
Now, one technique that's really revolutionized structural biology more recently is cryo-EM, isn't it?
Oh, absolutely. Cryo-electron microscopy has been transformative, especially for determining the structures of large protein complexes. It even snagged a Nobel Prize in 2017.
What makes it so powerful?
Its big advantage is how it prepares the sample. Traditionally, to get atomic-level detail, scientists often had to crystallize proteins to study them with X-ray crystallography. But getting crystals can be incredibly difficult, impossible for many large or flexible complexes, and the crystal structure might not perfectly represent the protein's natural state.
So cryo-EM avoids crystallization.
Yes. With cryo-EM, you take your sample, say, purified ribosomes or viruses in solution, and you freeze it extremely rapidly, so fast that water molecules don't have time to form damaging ice crystals. They get locked in a glass-like state called vitrification.
Preserving the structures in their native state.
Exactly. The proteins or complexes are frozen in various orientations, just as they were in the solution. You then use an electron microscope to take tens or hundreds of thousands of images of these frozen particles. Powerful computer algorithms then sort these 2D images by orientation and average them together to reconstruct a high-resolution 3D map of the molecule.
Getting close to atomic detail.
In many cases now, yes, near atomic resolution. This lets us see precisely how these complex molecular machines are built and how they might work.
And this isn't just academic anymore. It has real-world impact.
Hugely so. Cryo-EM was absolutely instrumental during the recent pandemic, for example. Researchers used it incredibly quickly to solve the detailed structures of the SARS-CoV-2 spike protein and other key viral components.
Which helped with vaccine design.
Massively. Understanding the exact shape of the spike protein allowed for the rapid development of vaccines that target it effectively, and also helped in designing potential antiviral drugs. It's moved from a specialized technique to a frontline tool in biomedical research. We can literally see the machinery of life and disease in action, frozen in time. #tag outro. So if we were to wrap up this whole tour, I think the biggest takeaway is seeing the cell not just as a collection of parts, but as this incredibly integrated system. It's defined just as much by its dynamic processes, things moving on the cytoskeleton, vesicles trafficking between organelles, as it is by its compartmentalized structure.
Yeah, it's the coordination that's key.
It is. The seamless communication and coordination between the nucleus, the ER and Golgi assembly lines, The lysosomal recycling centers, the mitochondrial power plants, all orchestrated by that internal transport network. That's what allows cells to adapt, to respond, and ultimately to build complex tissues and organisms like us.
And the implications for us, for health and medicine, are just immense, aren't they? Every detail we've covered, whether it's topoisomerase letting DNA replicate, or E-cadherin holding tissues together, or how lysosomes recycle waste, it's not just textbook stuff.
Not at all.
It is the difference between health and disease. This is the level we need to understand to design targeted drugs, to fight infections effectively, to understand aging, to tackle cancer.
Yeah.
It's all happening at this microscopic, incredibly complex level.
Understanding the cell truly is understanding the foundation of all life. And maybe as a final thought for you to chew on, we talked quite a bit about endosymbiotic theory.
Right, the idea that mitochondria and chloroplasts used to be free-living bacteria.
Exactly.
Yeah.
Evident because they still have their own bacterial-like DNA and 70S ribosomes. Think about your mitochondria, these essential powerhouses in almost every cell in your body. The organelles that provide your energy but also hold the keys to initiating cell death through apoptosis.
Yeah.
Consider the fact that these absolutely critical components of you have a completely separate evolutionary history from the rest of your genome, inherited only from your mother, tracing back over 1 billion years to an ancient bacterium that took up residence inside another cell. That tiny organelle is a living relic of arguably the most important symbiotic merger in the history of life on Earth. It's still there, inside you, right now.